Hello and welcome back to the Bio 181 series on metabolism. In this lecture, we will discuss the major form of cellular energy, adenosine triphosphate, or ATP. We will also discuss the cellular mechanisms of its generation. First, it is important to understand what ATP is. Recall that when we talk about energy in this class, we are often referring to energy in the form of electrons stored in chemical bonds. ATP is a molecule composed of adenosine, a nucleotide, bound to a sugar called ribose, which is bound to a series of phosphate functional groups. Due to the nature of the bonds in this molecule, when there are three phosphate groups attached to the ribose, the bond between the second and third phosphate is considered a high-energy bond. Essentially, the electrons in this bond are ready and willing to pop out of there and help facilitate other chemical reactions. When there are three phosphates attached to the ribose, this molecule is called adenosine triphosphate. Tri, because of the prefix tri means three. When we break this bond and a phosphate comes off, this molecule is left with two phosphates, and thus is called adenosine diphosphate, or ADP. When another phosphate pops off, which can happen, the molecule would be left with one phosphate, and thus it would be called adenosine monophosphate, or AMP. However, ATP with three phosphates is the most energetic of these three due to the high energy bond between the second and third phosphate group here. Because of this, ATP is the preferred source of energy in cells. A benefit of ATP being the preferred source of energy in cells is that it functions sort of like a battery in that it can be charged up. ATP stores a lot of energy in that high energy bond. But when that bond is broken and energy is released to perform other cellular work, this results in the lesser energetic adenosine diphosphate, or ADP, plus the phosphate that popped off. However, we can recharge the ATP battery if we invest energy in recreating the bond between ADP and this phosphate. Anytime we add a phosphate group to something, it's called phosphorylation. Thus, when we attach a third phosphate group to the ADP to make ATP, we can call this process phosphorylation. Again, building bonds requires energy, and this includes building the bond between ADP and this phosphate. There are three primary mechanisms of generating ATP in a cell. These are primarily distinguished based on where the energy that we are investing in the bond between ADP and the third phosphate is coming from. The first method is substrate level phosphorylation, or SLP. In this method of ATP generation, the energy is gained from an exergonic reaction. The phosphate is actually gained from a molecule involved in this reaction. Recall that when we talk about chemical reactions, substrates enter a reaction and products leave a reaction. So substrate level phosphorylation is so named because the phosphate is obtained from a substrate during an exergonic reaction that generates enough energy to create ATP. This process does usually involve an enzyme that enables the transfer of the phosphate. The second method of phosphorylation is called oxidative phosphorylation. This method of ATP generation is a little more involved because it requires the freedom of electrons from the chemical bonds and subsequently using those electrons to establish a concentration gradient. The concentration gradient is the source of energy that phosphorylates ADP and creates ATP. The third method of phosphorylation is called photophosphorylation, and as you can probably infer, involves the use of light to generate ATP. In this case, instead of enzymes releasing electrons from chemical bonds, as is the case with oxidative phosphorylation, energy from light, or photons, release electrons from chemical bonds. However, similar to oxidative phosphorylation, these enzymes are subsequently used to generate a proton motive force, which is a source of energy for the generation of ATP. Again, these three methods of making ATP are distinguished by where the energy that is used to create the bond between ADP and the phosphate group comes from. Either an exergonic reaction, as with substrate level phosphorylation, or a concentration gradient, as with oxidative or photophosphorylation. Oxidative phosphorylation establishes a concentration gradient using electrons collected from enzymatically broken bonds, while photophosphorylation establishes a concentration gradient using electrons from bonds broken via light energy. Let's look at each of these individually and a little more closely. Let's start with substrate level phosphorylation. To orient you to this slide, you see a molecule with a bond. One of the monomers of this molecule has a phosphate. In substrate level phosphorylation, an enzyme breaks a bond in this molecule. Recall that catabolic reactions are exergonic, so energy is released from this bond breaking. This is enough energy to fuel the addition of a phosphate to ADP to create ATP, the phosphate of which was also released from the substrate of this reaction. Terminology will be your saving grace here. Recall that a substrate is something that enters a chemical reaction. In this case, this is the substrate, and it has a phosphate. The substrate is involved in a catabolic and exergonic chemical reaction where bond is broken and energy is released. This is the energy that is used to fuel the addition of a phosphate to ADP to create ATP. The phosphate in this case is also obtained from the substrate. As mentioned, SLP is fueled by enzymes. This is showing that process. You can see that this substrate has a phosphate attached to it, and the enzyme is actually what transfers this phosphate to ADP to create ATP. Okay, now on to our next method of ATP generation, oxidative phosphorylation. Oxidative phosphorylation, or OXPHOS, involves a series of catabolic and exergonic reactions that end up releasing a bunch of electrons. These electrons are harvested by molecules called electron carriers, which also bind up hydrogen ions, which are positively charged and can balance out the negatively charged electrons, making these electron carriers uncharged when they carry an electron, which is important for homeostasis anyway. These electron carriers carry the electrons and hydrogens to a membrane, release the electrons and hydrogens, and the electrons generate a concentration gradient of hydrogens, which is used as a source of energy to add a phosphate to ADP to create ATP. So again, terminology is really important here. We understand that phosphorylation is referring to a phosphate being added to ADP to create ATP. But why oxidative? Well, in chemistry, oxidation is the term for a reaction in which an electron is lost from a substrate. So when a bond is broken or catabolized and an electron is lost from that bond, the substrate is said to have been oxidized. Since during oxidative phosphorylation, bonds are broken to release electrons, which are subsequently used to generate a concentration gradient, we call this oxidative phosphorylation, referring to the fact that the energy used to establish the concentration gradient is gained from oxidation or release of electrons from molecules. So oxidative phosphorylation is a two-step process. First, a molecule is oxidized, meaning electrons are released from a bond within the molecule. These electrons are picked up by molecules called electron carriers. When a molecule picks up or receives an electron, it is said to be reduced. This is because when a molecule gains an electron, it gains a negative charge, meaning its overall charge is reduced. So for step one molecule loses an electron or is oxidized, and another molecule gains an electron or is reduced. Reduced molecules often also pick up a corresponding hydrogen ion because these have a positive charge, which can neutralize the negative charge associated with the electron. Then these reduced molecules, electron carriers, carry the electrons and hydrogens to a membrane where they will generate a concentration gradient. But let's focus a little bit more on the first step. First, let's make sure we understand what oxidation and reduction are. As mentioned, oxidation involves a molecule losing an electron, while reduction involves a molecule gaining an electron. So in the above reaction, A is oxidized when it loses this electron, and B is reduced when this electron is transferred and received. Since it's not really ideal for random electrons to be floating around in space, oxidation and reduction reactions are always paired. If a molecule is oxidized and loses an electron, a corresponding molecule will be reduced and gain an electron. Since oxidation and reduction reactions are always paired, reactions like this are called redox reactions. There are a couple fun mnemonics that may help you remember what happens in redox reactions. The first, which is what I learned, is oil rig. Oil stands for oxidation is loss, and rig stands for reduction is gain. Again, this may seem confusing, but just think about the fact that gaining an electron would result in a reduced charge. Another mnemonic that I was taught by a colleague is Leo the lion says grr. This would be loss of electrons is oxidation and gaining is reduction. I honestly don't care how you remember it, just make sure that you do. Even better, don't just make sure that you remember it, make sure that you understand it. Okay, so this slide will hopefully better demonstrate redox reactions. Here you have a molecule, an enzyme breaks a bond in this molecule, and when that bond is broken, an electron is released. So this molecule is oxidized. Another molecule, in this case molecule X, accepts the electron and becomes reduced. To balance out the negatively charged electron, X also binds with a hydrogen ion. So the reduced compound would look like XH. We will discuss cellular respiration in future lectures, but several reactions involved in this metabolic pathway are excellent examples of redox reactions. During cellular respiration, glucose is catabolized, but it is actually oxidized because its catabolism results in the loss or release of electrons. These electrons reduce electron carriers. The ones associated with cellular respiration are NAD and FAD. When these accept the electron from glucose's oxidation, they become reduced. To balance out this negative charge, they also accept a hydrogen ion. Thus, you can identify if an electron carrier is carrying an electron if it has a hydrogen. For example, if NAD has become NADH, you know it's carrying an electron. Anyway, we're going to discuss more about this later. So what happens after a molecule is oxidized and a corresponding molecule, an electron carrier, is reduced? Well, the electron carrier then serves the function of carrying that electron and the corresponding hydrogen to a membrane, where it will use the energetic electrons to establish a concentration gradient of hydrogens, which will then provide the energy necessary to phosphorylate ADP into ATP. Hydrogen ions are hydrogen atoms that have lost their electrons. The atomic number of hydrogen is 1, and they have no neutrons. So when they are, because they're literally like one proton, they're sometimes referred to as such. So when a concentration gradient of hydrogen ions is created, this is sometimes referred to as a proton motive force, or PMF. The electron transport chain, which is the terminal step of cellular respiration, is a great example of a proton motive force being established as a result of redox reactions. Throughout cellular respiration, glucose is oxidized, releasing electrons from its bonds being broken. These electrons are picked up by electron carriers, such as NAD and FAD, along with corresponding hydrogen ions. These carriers carry the electrons and hydrogens to a membrane, such as the crista of mitochondria. Once the carriers get to a membrane, they release the electrons and hydrogens. The electrons are used as energy to fuel the pumping of hydrogens to one side of the membrane. Eventually, this leads to the formation of a concentration gradient, a ton of hydrogens on one side of a membrane. Recall that concentration gradients are a robust source of cellular potential energy. This is the energy that can be used to phosphorylate ADP into ATP. When a molecule's concentration gradient is used as energy to fuel a reaction, this can be referred to as chemiosmosis. I know this is so annoying, so many terms for the same thing. Just recall that a concentration gradient is when there are more molecules on one side of a membrane compared to the other. And proton motive force is when this is a concentration gradient specifically of hydrogen ions, which again are sometimes just referred to as protons. And chemiosmosis is when a concentration gradient is used as energy for another reaction. The electron transport chain is an example of all of these. So to summarize, oxidative phosphorylation is when a molecule is oxidized, releasing electrons. These electrons are bound up along with corresponding hydrogen ions by electron carriers. These electron carriers carry the electrons and hydrogens to a membrane. At the membrane, the electrons are used as energy to generate a concentration gradient of hydrogen ions, or a proton motive force. This is energy in the form of chemiosmosis, which fuels the phosphorylation of ADP into ATP. So 2 steps: redox reactions and a proton motive force. There is something that I want to note about proton motive forces. Chemiosmosis of hydrogen ions is able to provide enough energy to phosphorylate 1 ADP into 1 ATP per hydrogen ion. So this means that for every hydrogen ion pumped across the membrane involved in the proton motive force, 1 ATP will be generated. In this image, you see 1, 2, 3, 4, 5, 6, 7, 8 hydrogen ions on this side of the membrane. This means that 8 ATP will be generated. This will be important primarily later, but I just want to mention it now as it pertains to the function of proton motive forces. Finally, we can make ATP using energy from the sun. This is called photophosphorylation. This process is actually quite similar to oxidative phosphorylation, but instead of an enzyme oxidizing an organic compound, light energy is used to oxidize a water molecule. This does release electrons and hydrogens, and these are used to generate a proton motive force, which leads to the phosphorylation of ADP into ATP. We will discuss photophosphorylation later in greater depth. For now, just know that energy from the sun is what enables the establishment of a proton motive force, and this is what enables phosphorylation of ATP during photophosphorylation. Okay, great. So now you know that we can make ATP in 3 ways. 1, we can generate ATP using energy released from exergonic reactions via substrate level phosphorylation. In this process, the phosphate group is also obtained from the exergonic reaction. 2, we can generate ATP by releasing electrons via oxidative reactions and carrying these electrons using reduced electron carriers to a membrane, where the electrons will then be used to establish a proton motive force, which provides enough energy to phosphorylate ADP into ATP. Or we can generate ATP by using light energy to release electrons from water, and using these electrons to generate a proton motive force, which will provide the energy for phosphorylation of ADP into ATP. These are important mechanisms by which ATP is generated. In the next few lectures, we will talk about cellular respiration, which can generate a ton of ATP using SLP and OXPHOS. Then next week, we'll talk about photosynthesis, which can generate some ATP via photophosphorylation. It's important that when you see a reaction that generates ATP, that you can identify which of these methods was utilized. Make sure that from this lecture, you understand the basic structure of ATP, ADP, and AMP, that you understand how ATP functions similar to a rechargeable battery, that you're able to differentiate SLP, OXPHOS, and photophosphorylation, that you understand what happens to a molecule when it is oxidized or reduced, and that you understand how concentration gradients can provide energy via chemiosmosis. You need to know the terms ATP, photophosphorylation, substrate level phosphorylation, oxidative phosphorylation, photophosphorylation, oxidation reduction, redox reactions, proton motive force, and chemiosmosis. Thank you so much for watching this lecture. You are doing phenomenal. Keep it up.